My name's Steve Jackson. I'm based in a place called the Gurdon Institute, which is at the heart of Cambridge, UK. In the Gurdon Institute, we're trying to understand cellular and molecular biology as it relates to cancer and also development, and to try and translate the insights that we obtain in these arenas towards understanding of human disease. So today, what I'm going to share with you is a little bit of a personal perspective of the last 21 years, and at the end, thinking about what the next 20 years or so might hold for my research, if I'm still going that long, or research in the field that I'm in. Now, as a backdrop, I just wanted to highlight the field that I'm in and explain its relevance at the cellular level and also at the level of understanding human disease. So it turns out that every human body, so you, have around 10 to the 13 to 10 to the 14 cells in your body. So that's 10 with 13 or 14 zeros, somewhere between that, in a single human body. And each cell in the body contains billions of base pairs of DNA. Now, DNA is a fairly inert chemical. It doesn't rapidly decompose, but the DNA in our cells is continually being bombarded by DNA damaging agents. Some of these are coming from the environment. These could be ionizing radiations, ultraviolet light, toxic chemicals coming from the environment. But actually, much of this damage comes from endogenous sources. Our intrinsic metabolism generates chemicals which damage DNA. So consequently, our cells are having to deal with tremendous amounts of DNA damage. Every cell in your body is damaged around 100,000 times per day. So our cells, and in fact, every cell on Earth, has evolved systems to repair, detect the presence of, repair, and signal the presence of DNA damage. And we know that now that there are a whole host of proteins in our cells which detect the DNA damage and mediate these DNA repair and associated processes. And in fact, it was through stumbling across one of those, a protein called Ku, about 25 years ago, that my lab got into this whole arena. It turns out that the Ku protein basically behaves as a molecular policeman. It surveys the cell, the nucleus of our cells, looking for DNA damage, not normally doing very much, but when damage occurs, the Ku protein binds to that damage and sets off the emergency and the repair services. So it turns out there are many different forms of DNA damage and many different proteins dedicated to different forms of DNA damage. Now, these processes are very important for the function of our cells, but we also know they're medically important because acquired or inherited defects in these processes, DNA repair and associated signaling and other events in the cell, give rise to various diseases. And these can be acquired or inherited defects giving rise to these conditions. So conditions caused by DNA repair defects range from premature aging, developmental defects, neurodegeneration, premature aging, reduced fertility, and cancer. And in fact, my lab and many others in the field now are particularly interested in cancer, which is generally caused by accrual of mutations in a particular cell, which sets it on the path to be cancerous. But we're also interested in this connection to aging, both normal aging and premature aging, which is probably caused by accumulation of damage in somatic cells of the body. So clearly, understanding these DNA repair processes should provide insights into certain diseases. But over the years, my lab and many others in the field have been interested in trying to exploit that knowledge to come up with ways to improve people's health. Well, with that as a backdrop, in almost exactly 21 years ago now, in fact, I decided, with the help from Cancer Research UK and Cambridge University, to set up a company. And this company was called Kudos, because my whole career is based on what the protein Ku does, this DNA binding molecular policeman protein. And the bold aim of Kudos was to develop small molecule inhibitors, drugs if you like, that inhibit DNA repair and associated processes. And I was very excited about this opportunity because I could see how it could be used in many different disease areas. But it turned out to be not quite so straightforward. So here was this young professor from Cambridge talking about setting up a company to develop DNA repair inhibitors, but pharmaceutical companies were not interested and venture capital groups were not interested. Why is this? Well, it was counterintuitive. Why would you want to inhibit DNA repair with drugs when defective DNA repair gives rise to disease? So it actually took me almost 2 years of dedicated effort reworking my presentations and thinking about things, how I could pitch it in the most clear way to potential investors. And in the end, I was successful, and Kudos got started, and over a number of years, developed into a small biotechnology company just outside Cambridge. Now, one of the key concepts behind Kudos was using DNA repair inhibitors to target cancer. So how can we target cancer with DNA repair inhibitors? Well, it's worth just stepping back for a moment and thinking what cancer therapy or a successful cancer therapy actually does. To be successful, a cancer therapy has got to kill the cancer cells or stop them growing, but not kill the normal cells or stop the growth of normal cells of the body. And the only way you can really achieve that is by understanding and exploiting a difference between the cancer cells and the normal cells. Now, we've known for many, many years that many cancers lose DNA repair pathways during their evolution. So a key difference between most cancers and normal cells of the body is that the cancer is defective in DNA repair, the normal cells are proficient in DNA repair. So that's one concept that was in the background. The other concept is a genetic concept that's often referred to as synthetic lethality. Now, it's a very complicated term, but what it really means is that our cells, in fact, all life forms, have evolved to be robust. And that often means that our cells have multiple ways of doing certain essential processes. And our cells have backup pathways when one pathway fails. That makes a lot of sense. Well, one of the ideas is that if a cancer cell has lost a backup pathway, then it's going to be very reliant on a particular pathway, whereas the normal cells would have more redundancy and more robustness. So translating that into oncology and in terms of a therapeutic principle, this synthetic lethality principle, which means you can lose the function of gene A and be fine, you can lose function of gene B and be fine, but you can't lose both, can be translated into a pharmacological application. So one of the concepts behind Kudos was to basically develop drugs, in this case, against the product of gene A. So the speculation would be, if you inhibit gene A in normal cells, you won't kill normal cells because they have the other pathway B. But what would happen, for example, if a cancer cell lacked that pathway B? So to cut a long story short, Kudos developed inhibitors of various DNA repair enzymes, one of which is called PARP. This stands for poly-ADP ribose polymerase 1 and 2. And basically what we found with our colleagues is that PARP inhibitors are not toxic to normal cells, but they are toxic to cancer cells lacking pathway B, in this case BRCA1 or BRCA2. Okay. So several years later, drugs developed by Kudos and formulated by Kudos were taken into and through the clinic by AstraZeneca, and this led to the clinical success of Lapparib, trade name Lynparza, for the treatment initially of BRCA1 and BRCA2 mutated ovarian cancers, but now also ovarian cancers without BRCA1 or BRCA2 mutations and other tumors such as breast tumors, breast cancers. So it turns out that Lapparib was the first DNA repair inhibitor drug marketed worldwide. If you like, it's the world's first synthetic lethality medicine. It's the first cancer drug that's been launched targeting an inherited predisposition. It was also notable that this drug was launched with a genetic diagnostic. So if you like, Lynparza or Lapparib is one of these precision medicines that we've all heard about, in this case specifically targeting a particular protein, in this case PARP, in particular contexts, the context of BRCA deficient patients, to selectively treat those tumors. As I mentioned, Lapparib and other PARP inhibitors have also now been licensed for other cancers, and I think there's a lot of excitement that that could be broadened yet further. Now, from a personal perspective, it's wonderful to know that this drug, which originated out of basic cell and molecular biology research in my lab and my collaborators' labs, and through Kudos and AstraZeneca, has now gone into thousands upon thousands of patients throughout the world, many of which are benefiting. And it's also good to know that finally, after many years, DNA repair has really caught on. There are many pharmaceutical companies now with DNA repair inhibitor programs, and also various biotech companies being launched throughout the world in the last few years to exploit this and other opportunities for DNA repair, not just in oncology, but in other therapeutic arenas. Now, from my perspective, I'm not focused on the financial side. Obviously, drugs like Lapparib are potentially going to make AstraZeneca and Merck, which are co-developing this compound, lots and lots of money. The drug was recently valued at around $18 billion. I don't get any of this as royalties, by the way, so my excitement stems from the fact that some patients are actually benefiting from this. And it was wonderful for me a couple of years ago, when Worldwide Cancer Research a charitable organization which funded some of my early science, together with Cancer Research UK, basically did a video. They videoed me getting up one morning, and at the same time videoed a lady who'd been treated successfully with the Lapparib, and arranged for us to meet in the Botanic Gardens in Cambridge for the first time. It was quite an emotional event, learning about this lady who was initially going onto a Lapparib trial with no longer responding to existing therapies. And 8 or 9 years now on, this patient is totally tumor-free. And she was talking about the fact that she'd seen her daughter's wedding, she'd been to Australia and done all these things. I met her husband too. So for me, that is probably the most meaningful outcome of my entire scientific career. Okay. But let's not get too overrun with this. That is a success story. Many patients are responding to PARP inhibitors of various kinds throughout the world, but some patients aren't, even some patients with BRCA1 and BRCA2 mutations. So I think a major challenge now is to understand how some tumors, even with BRCA1 and BRCA2 mutations, are not responding, and how in other cases, a patient whose tumor initially responds may evolve resistance. So one key goal for my lab right now is to try and understand these resistance pathways, to try and systematically exploit these pathways and ideally translate that knowledge towards clinical applications. So we are actually at a very exciting time now in science. I've been in science for over 30 years as a researcher, since starting my PhD. It's always been exciting, but I think the next 20 years promises to be even more exciting. The reason for this now is that we have at our fingertips techniques and technology and knowledge that we wouldn't have deemed possible just a few years ago. So next generation DNA sequencing developments in that arena now mean that we have insights into genetic diseases, cancer, and a whole range of other diseases. We also have methodologies such as iPS cells, CRISPR-Cas9. We can do things now that we wouldn't have dreamed possible just a few years ago. So using some of these tools, for example, my lab has recently found mechanisms of resistance to Lapparib and other DNA repair inhibitor drugs. And one of the things that's really exciting is at the moment is that what we're finding is that the resistance pathway for one drug is providing a vulnerability, an Achilles heel for another drug. And that really, I think, opens up the prospect that a disease like cancer, if we can understand resistance pathways to new and existing drugs, we might be able to anticipate them, possibly stop them occurring. In other cases, we might be able to actually use those resistance pathways. That's another change that cancer has made, targeting the resistance pathways to kill the cancer via other mechanisms. So I'm optimistic in the long term. Of course, cancer is not going to be cured in any time soon, but I think with this increasing type of knowledge, we're going to be increasingly be able to chase cancer into blind alleys. And when we're thinking about DNA repair processes, these are fundamental processes. There are only certain ways you can do it. So if we can anticipate resistance pathways, block them off, or take advantage of them, I'm very confident that we've got an exciting future ahead of us in this arena. So just to finish up, I just want to step back for a moment and say, yes, my lab for the last 25 years has been focused mainly in the cancer arena. But these same tools, these methodologies that I've been talking about, also apply to a whole range of other diseases. Health and disease, it's all about balance and imbalance. Disease is associated with imbalanced cellular or intracellular processes. Now, in the context of cancer, what we're trying to do is identify an imbalance in cancer and tip the cancer cell over the edge into death or permanent growth arrest. But I think there's also exciting opportunities thinking about things the other way, something that we call synthetic viability. If we can have a disease cell, if we can understand the imbalances in that disease, I think there's going to be some very exciting opportunities to identify ways of rebalancing disease physiology, to rebalance diseases, genetic diseases, and other diseases. And so with this type of paradigm over the last few years, my lab has recently identified potential therapeutic strategies for a whole range of conditions, ranging from a neurodegenerative disease, a hematological disease, premature aging, and also another form of cancer. So I personally am very, very optimistic and excited about the next 20 years, and I'll be happy to discuss those kind of opportunities with you in questions. Thanks.